Yeah, thank you so much, Alice, for those powerful insights into the impact of metabolomics and the exciting case studies. Now that we've seen how metabolomics can drive breakthrough discoveries, it's time to show you how we at biocrates are turning that potential into reality with our newest essay, MxP Quant 1000. In the second part of this webinar, I will walk you through how Quant 1000 makes the difference and sets a new standard in metabolomics. I will show the essential features that define the assay, what makes this assay stand out in terms of metabolite coverage and biological relevance, and how MxP Quant 1000 meets the diverse needs of different research fields. Metabolomics has become an essential tool for understanding health, disease, and biological mechanisms, but not all metabolomics services are created equal. Many solutions fall short when it comes to comprehensive coverage, biological relevance, precision, or efficiency. And that is where MxP Quant 1000 makes the difference. We have designed this service to give researchers the widest, most precise, and most insightful metabolomics data, all from the smallest possible sample volume. And here's how we set the new standard of excellence in quantitative metabolomics. MxP Quant 1000 offers one of the most extensive metabolite panels available, covering hundreds of metabolic pathways, what enables a holistic view of metabolic health and disease. It includes metabolites specific to the microbiome host axis, allowing researchers to investigate the interplay between gut microbiota and systemic health. By addressing both core metabolic functions and microbiome-related pathways, the assay supports research that connects fundamental biological insights with potential applications in chronic disease mechanisms and preventive strategies. Having access to individual metabolite concentrations is valuable, no question, but true biological insight comes from understanding how metabolites function together in the context of disease. That's why we go beyond just measuring metabolites. Our approach includes defined disease-specific metabolism indicators, what is unique in the market. So what makes this a game changer? The assay is pre-configured with biologically relevant sums and ratios that offer you stronger scientific value with immediate insights and without the need for extensive post-analysis calculations. These sums and ratios that we call metabolism indicators are designed to facilitate comparisons across different groups or patient cohorts. And this enables a deeper understanding of metabolic dysregulation in specific conditions like cancer or Alzheimer's disease, as demonstrated by Alice. That provides researchers and clinicians with interpretable and actionable metabolic data, improving biological interpretation and reducing the time from analysis to discovery, and this accelerates making decisions. That was biology. Let's talk about analytical performance. The assay incorporates hundreds of carefully selected calibrators and internal standards that ensures the highest level of quantification precision. This high degree of precision makes it ideal for biomarker discovery and validation, paving the way for translational research applications and to transform medicine. The assay has the best sample volume to data points ratio. That means maximum insights from minimal sample. With only 40 microliters of sample, the assay provides a comprehensive metabolic profile of over 1,800 biomarkers. And with this, ladies and gentlemen, we are setting a new benchmark in metabolomics. This makes it ideal for precious samples in clinical studies and biobanking, where sample volume is often a limiting factor. Another benefit here is cost efficiency. Quant 1000 reduces the need for multiple assays by delivering broad metabolic coverage in a single assay, optimizing for both time and resources. Other service providers split their assays to achieve comparable coverage, but this requires more sample volumes, usually several hundreds of microliters, and more assay preparations and instrument time, what makes it less cost efficient. So with MxP Quant 1000, you no longer have to compromise between coverage, precision, biological relevance, and efficiency. We combine the most comprehensive metabolite panel and disease-relevant insights with unmatched analytical precision and sample efficiency, all in a single validated service. Whether you're working on biomarker discovery, disease research, or fundamental metabolism research, our service ensures you get the most reliable and actionable metabolomics data available today. That's the difference we bring to your research. So, what are the exciting features of MxP Quant 1000? And I'm sure you're going to love this part. With 1,233 metabolites across 49 classes, we have significantly expanded our metabolite portfolio compared to our previous assays, predominantly on the small molecule side. The assay has been validated with human plasma and urine and requires a sample volume of only 40 microliters. On top of the 1,233 metabolites, you get 648 predefined metabolite sums and ratios with biological or clinical relevance, all based on published scientific research. This allows researchers to explore advanced insights into metabolic processes and pathways to gain deeper biological interpretation of the result. For an even more streamlined interpretation, the indicators are categorized by specific disease indications. This coverage of metabolites and its sums and ratios results in 1,881 biomarkers. At every measurement can be a metabolite or a ratio or a sum of metabolites is a potential biomarker. With this, we are reaching a new era in targeted quantitative metabolomics to study microbiome-host interactions, metabolic health, and disease metabolism. And this empowers researchers to explore hundreds of metabolic pathways and to contribute to the development of innovative solutions that will transform medicine. So with MxP Quant 1000, we are not just expanding metabolomics, we are transforming medicine with metabolomics. Now I would like to resolve these numbers. So let's take a look on the metabolite classes and numbers. On the left, you find the small molecules. On the right, the lipid panel. Compared to our previous MxP Quant 500 or Quant 500 XL assays, we have dramatically expanded the small molecules panel from about 100 metabolites across 14 classes to now 327 metabolites across 24 classes in Quant 1000. The classes highlighted here in yellow were expanded with more metabolites compared to Quant 500. The green ones are new biochemical classes. I would like to highlight a few of them that showed up in the 3 case studies presented by Alice. For example, we have increased the number of indoles and their derivatives. These are key microbial metabolites derived from tryptophan that influence gut health and systemic metabolic regulation. So this enhancement improves research capabilities in microbiome studies as presented by Alice. We have also increased a lot on the classes that feed into the TCA cycle and or are intermediates, such as fatty acids, carboxylic acids, or the new class of dicarboxylic acids. So with this assay, ladies and gentlemen, we now provide nearly complete TCA cycle coverage that allows for more in-depth and accurate metabolic profiling. And this is really exciting as this offers new insights into mitochondrial function, energy metabolism, and neurodegeneration, as shown by Alice in the case study of Alzheimer's disease. As a last example, the assay now includes a dedicated panel for polyamines that enables researchers to study their role in tumor metabolism and potential therapeutic targets. This addition makes the applicability of the assay in oncology much stronger, supporting biomarker discovery and cancer metabolism research. The lipid panel on the right side is similar to our previous products and covers 906 lipids across 25 classes. When you see all these incredible numbers, you might be wondering, how does this compare to untargeted approaches? So please allow me to elaborate on this. In contrast to untargeted approaches, where the scope is on identification of new metabolites using spectral libraries and on relative quantification of those, the targeted approach of Quant 1000 assay uses specific mass transitions of well-characterized metabolites for identification and absolute quantification. So we are not just measuring anything. Each metabolite is relevant and handpicked, specific for certain applications or diseases. Untargeted solutions can screen and identify several thousand of metabolite features, but untargeted data contains many features with unknown IDs that might or might not be regularly present, leading to a maximum number of identified compounds that is often around the same number of metabolites quantified by comprehensive targeted assays. In other words, a comprehensive panel of Quant 1000 can offer actionable, equivalent, or even higher coverage that is usually reported by untargeted methods. And this is our value proposition. The targeted quantitative method of Quant 1000 gives higher level information by focusing on defined metabolites relevant to biochemical pathways. This extensive coverage ensures that whatever your focus is, you have the right tool to uncover meaningful insight. With Quant 1000, you get one solution that meets the needs of nearly every research area, from fundamental research and clinical research to drug development or consumer health. In fundamental research, scientists aim for biomarker discovery or need comprehensive insights to understand the biological mechanisms underlying chronic diseases. With Quant 1000, you can do that. If the focus is on systems biology approaches, integrating metabolomics with genomics, transcriptomics, and proteomics, or if the interest is in gut microbiota and its role in disease, Quant 1000 can do that because the assay provides a complete picture of metabolic changes associated with chronic diseases or microbiome host interactions. In clinical research or pharmaceutical industry, early-stage biomarkers are a key focus area for understanding disease mechanisms and identifying potential therapeutic targets. Quant 1000 helps researchers here investigate metabolic signatures that may be relevant for disease risk stratification and precision medicine approaches. By analyzing metabolite sums and ratios, Quant 1000 enables a deeper understanding of metabolic alterations that could contribute to disease progression and treatment response. For pharmaceutical research, Quant 1000 supports the study of drug metabolism and the investigation of metabolic pathways linked to therapeutic response that can help optimizing dose selection and intervention strategies in a research setting. Consumers today are more proactive about their health than ever, seeking ways to monitor metabolic health before disease develops. While consumers don't use our products directly, routine labs and healthcare companies, they rely on our products to bring them reliable screening solutions. Also, the demand for at-home sampling solutions is growing, emphasizing convenience, accessibility, and early detection. Quant 1000 enables early detection of metabolic imbalances allowing healthcare providers to identify risks before symptoms arise and help consumers take preventive action. So once more, with MxP Quant 1000, you can do this. So no matter what your research focus is, fundamental science, clinical applications, pharmaceutical development, or consumer health, Quant 1000 provides the precision, coverage, and insight you need to make a difference. In the second part of my presentation, I would like to talk about our complete service portfolio with MxP Quant 1000 now added, about our modular offerings of Quant 1000, and beyond that, how you benefit doing metabolomics with us, from project design to data interpretation and up to multi-omics data analysis if desired. At biocrates, we offer a comprehensive portfolio of metabolomic solutions designed to meet the diverse needs of researchers across disciplines. Whether you need broad metabolic profiling or targeted analysis of specific metabolite classes, we provide the right tools to generate high quality and biologically relevant data. For researchers focusing on specific metabolic pathways, we offer specialized solutions tailored to defined metabolite classes such as bile acids, oxysterols, or short-chain fatty acids. Our profiling solutions provide wide metabolic coverage for those who need more global insight into metabolic pathways. With MxP Quant 1000, we are now not just expanding our portfolios, we are setting a new standard for metabolomics research. With Quant 1000, we provide a complete metabolomic solution integrating both small molecules and lipids into a single powerful assay. However, metabolomics research is diverse, and different studies require different levels of depth and focus. That's why Quant 1000 is designed with a modular approach, what you see here as MX Quant and LX Quant, allowing you to choose the components that best fit to your specific needs. Before I introduce you to these two modules, I want to give you a quick explanation here on the metabo indicator numbers of the new assays. Some of you might have noticed that the number of metabolism indicators in Quant 1000 does not directly match the sum of indicators from MX Quant and LX Quant. The reason for this is that 23 of the indicators in Quant 1000 are derived from the combination of both small molecules and lipids. These integrated indicators reflect biological relevance that emerges only when both analyte classes are considered together. Now let's briefly talk about the modularity. If your research is centered on small molecules, you can access this part of Quant 1000 panel through our analytical service. For example, who want to study the effects of the microbiome on the host, or more generally explore metabolism of small molecules, or screen for core metabolism. With the MX Quant assay, you will benefit from 327 molecules plus 291 indicators, resulting in over 600 biomarkers. Similarly, if lipid metabolism is your primary focus, whether for cardiovascular or neurological research, for example, you can choose to analyze only the lipids panel as part of our service in the LX Quant assay, which had a broad panel for circulating lipids from lipoproteins and membrane lipids. The LX Quant assay measures 906 lipids plus 334 indicators, resulting in over 1,200 lipid-based biomarkers. That was the pure measurement, but we also support you before and all the way to data analysis and interpretation, and multi-omics data analysis if desired. When you collect your samples, we can help you to design your study, and we can also help you after you have received your data, to understand the data better and maybe to think of strategies for data analysis. This includes our statistical package that is an additional item that you can order and discuss with us. What you would like to have exactly in terms of statistical analysis that is performed by us. In addition to the statistics, we would interpret the biology in the context of the experiments that you share with us. And since last year, we also provide multi-omics data analysis. And I would like to give this some momentum as well and encourage you to not only do Quant 1000 with us, but consider your samples for other omics. And then we can help to combine data from metabolomics with other omics with this service. This is powered by our partner Viogen, a company based in Europe that is specialized in bioinformatics. They have pipelines that process different omics specifically, so that each omic has its own special treatment that is based on the type of data. And then at the end, it integrates the different omic layers to help you see what are the changes that occurred in the different groups. So combining all the omics together, and for those who want to really dig deep into the meaning of that after Viogen has done the analysis, we can also do an interpretation project on top and then give you as many insights as possible on your metabolomic study, on your multi-omic studies. Toward the end, I would like to summarize what you get from us when doing metabolomics with us. Biocrates' mission over the last 20 years has been to lower the entry barrier into metabolomics. Metabolomics holds incredible potential, but scientists still face significant roadblocks that limit their ability to fully benefit from its power. Going clockwise in our colorful donut here, starting with expertise, that remains a major bottleneck for many project teams. Metabolomics requires deep knowledge of analytical chemistry, biology, and bioinformatics, but not every lab has those specialists in-house. At Biocrates, our team of experienced experts is here to support you at every step, from study design to sample measurement, data analysis, and even data interpretation, to ensure you get the most out of your metabolomics research. Many labs lack the funding of high-end metabolomics infrastructure, from expensive mass spectrometers to skilled personnel. That's why Biocrates offers a cost-effective outsourced solution, giving you access to world-class metabolomics without the heavy investment in equipment and staffing. Variability in sample preparation, analytical methods, and instrumentation are well-known pitfalls in metabolomics. At Biocrates, we have developed highly standardized technology and workflows with rigorous quality control and validated methods that have enabled the implementation of standardized and reproducible metabolomics in population health studies, where thousands of samples are combined seamlessly, so you can trust your data every time. Metabolomics generates vast amounts of data, requiring advanced bioinformatics tools and expertise. Many researchers struggle to extract meaningful insights from their results. At Biocrates, we simplify this process with cutting-edge software and expert support, making high-quality data interpretation more accessible than ever. And in the final analysis, as just described, metabolites don't exist in a vacuum. They function together with genes, proteins, and other molecules. They can also be measured with omics and combined in multi-omics studies for deeper biological insight. At Biocrates, we have implemented an extra layer of our services to bridge the gap between metabolomics and other omics. So at Biocrates, we believe metabolomics should be a solution, not a struggle. That's why we have built a service ecosystem to provide a service that scientists can trust for reproducibility and expert-driven insights. Whether you are in academia, pharma, or clinical research, our solutions give you the confidence and expert support you will hardly find anywhere else. So we are really proud of our new solution because metabolomics is only as powerful as the technology behind it. And that's where MxP Quant 1000 makes the difference and those key challenges. It sets a new standard of excellence in metabolomics profiling because Quant 1000 delivers the most comprehensive quantitative measure of metabolism. Thanks to its high analytical precision, Quant 1000 delivers deep biological insights from just minimal sample volumes, making it a powerful tool for fundamental pharmaceutical and clinical research. So MxP Quant 1000 gives scientists the solution to decipher chronic disease mechanisms and identify phenotypic changes, contributing to the development of innovative solutions that are driving the transformation of medicine. As we come to the end of this session, we hope we have shown you how MxP Quant 1000 truly makes the difference and how you can benefit from partnering with us for your metabolomics research. If you have any questions or would like to explore how Quant 1000 or our other service offerings can support your work, we invite you to visit our website and we would love to hear from you. So let's continue this conversation. Today, we introduced the MxP Quant 1000 as an analytical service, but this is just the beginning, dear ladies and gentlemen. We also recognize that some laboratories prefer to run metabolomics in-house for greater flexibility and control. And I can already see many smiling faces out there now. That's why we're excited to announce an upcoming event. Later this year, Quant 1000 will also be available as a kit. Ready to use, standardized with all components you need, bringing the same level of precision and comprehensive coverage directly into your laboratory. If you're attending the ASMS conference this year, we would love to welcome you to our breakfast seminar, where we will officially present the kit version of Quant 1000. You will get a first look at its features, components, and what's needed to successfully implement it in your lab. We are also very happy to be joined by a guest speaker who will share preliminary data from a case study showing how the solution performs in real research. Thank you so much for joining us today. And now we are happy to take your questions.